BIO210L students. Welcome to lab. Today we have myself, Dr. Ju.
Me, Melissa.
And your tech, Jessica.
And we are all here maintaining our social distance, some of us in our protective gear as much as we feel comfortable. And we're going to be demoing the genomics lab today. And I know you guys are all going to read the worksheet that's online, so you'll understand the general pretext and context of what we're doing. Today we're going to focus on the first step in genome sequencing, and that is DNA isolation. There'll be a video online giving you some background information on this, so if you haven't watched it already, please watch it. And it'll tell you about the species that we're sequencing and about some of the other background about this effort. But today we're going to focus on just the DNA isolation part, which is, of course, is really important when we're trying to sequence DNA. We're going to be using some tools that people are using right now to target COVID-19, as well as lots of other really cool research. But we're going to isolate DNA. We're going to each take a little bit, since you don't get through the experiment. It was going to be a little bit of a competition between students, but now we're going to be getting a competition between us. And we're each going to make some changes to the protocol. And so, Jess, what change are you going to make to the protocol?
I'm going to keep our sample on ice the whole time.
And Melissa, what are you going to do?
So there's a few steps in the protocol that call for vortexing to mix the sample, but instead of doing that, I'm just going to very carefully invert and mix super gently.
And at the end, there's a part where we're going to use magnetic beads to isolate our DNA from our sample. I'm going to decrease the amount of magnetic beads and do an extra wash. So there's going to be a question on your worksheet, and you're going to have to answer why we are making these changes. So there are three areas that we want to target that could give us a bad DNA isolation result, or in other words, if we do a better job at it, we might get a better DNA result. And we're going to see if the changes we can make, who has the most effect on what we're doing. So, little healthy competition. We'll see who gets to win, and the winner gets their DNA sequence. All right. So let's start off by looking at some of the ways that we set up for the lab. So here's an example of a sample taken from a river in Mexico. Next, we are going to dissect out the muscle tissue from this fish. Once we have a good piece of tissue, we'll weigh it to make sure that it's within the right size. And then we'll take that piece of tissue, we'll put it in our salt buffering system and grind it up a little bit using a machine called a polytron. And then it'll, just real quick, about 30 seconds, then it'll be ready for to put in the incubator to be digested and isolated. Next, we prepped our flow cells for the sequencers so that they were ready to be used, and went off into the lab to get started on isolating our DNA for the next step. Let's see. Sample 1, sample 4. So first thing I need to do is add 300 microliters of 300 microliters of now we go wait. All right, our spin should be done. Let's go get our sample. Gonna avoid the pellet. Yeah, just guesstimate based upon the pellet is just cellular debris. So we're gonna try to avoid the pellet. Hopefully the DNA is in there. Cellular debris and salts. So that's about 500 microliters and a little bit more, maybe 550. So then I'm going to add an equal volume of isopropanol. I think it's about 550 in both cases. I'm just going to check with one of these pipettes. I'm going to set it to 550, and I'm going to see if it matches. Ooh. I still had some left, so there's more than 550. 650 was too much. Looks like it's just about 600. Because when I used the pipette, I didn't get any bubbles in it. So now I'm going to add 600 of isopropanol to both of these tubes. 100% isopropanol. And 100% isopropanol. Give it a little bit of a shaky, back and forth. Or you can flick it to mix gently. Now we're going to spin it again. We're going to now incubate it, in -20 for 10 minutes to get the DNA to precipitate out. So right now we're trying to salt out the DNA. So we lowered the dielectric constant of the solution, which should hopefully cause the DNA to precipitate out of solution. So let's go put it in the freezer. Gotta go get our stuff out of the freezer where it's been precipitating. I'm gonna put it in there, make sure the hinges are facing outward so we know where our pellet is, because we might not be able to see it this time. There's the pellet, it's just really small. Can't really see it very well. Keeping everything on ice. All right, now we need to take off, decant out the rest of the liquid. Yeah. Gotta avoid the pellet. And it's really hard to see, but there's a little pellet right in there. It's very faint. Because it's hard to see, we'll make sure that we pipette away from the pellet on the side away from the pellet. Still there. I don't know if you can see it or not, but there's still a little pellet there. You can see it right there. That is hopefully our DNA. So now I'm going to wash my pellet with 70% ethanol. And then we're going to spin it for 3 minutes at maximum speed. To make sure that the pellet stays in the tube. So now we've got smaller pellets. By the way, you might want to use the 200s to take it out, just because it's got a finer pipette tip. We'll cheat though. See if we can't make it work a little bit better. Yeah, that one's stuck to the side better. So we're going to pull away the ethanol away from the pellet, since the pellet's on this side. Ethanol's gone. Time to resuspend. 75 microliters of TE. And eventually dissolved. I'm going to spin it down real quick though to make sure that everything's at the bottom of the tube. So it's all dissolved, resuspended in water, and good. So now I'm going to add magnetic beads. And I'm going to add less than what the protocol says, because I want to try to target certain pieces of DNA and remove extra contaminants. So instead of 30 microliters, I'm going to add in 25. These are the magnetic beads. I'm going to pipe it up and down a couple times to mix the beads. And now I'm going to let the magnetic beads incubate with the sample for a minute. So after a minute of incubation with the beads, I'm now going to put it on this magnetic rack. And the magnetic rack is going to pull the beads towards the magnets, and the DNA that I want should all now be bound to the magnets. So I'm going to put it here. Notice how it's all brown across the whole thing. And it's hard to see, but right now all the brown beads are moving towards the magnet. So they're pelleting all of the beads away out of solution and towards the magnet. And all the DNA that I want is bound to the magnet. And all the contaminants and small pieces of DNA and things that I don't want are going to end up staying in solution. So I can extract them while I leave this thing on the magnetic rack, which will hold the beads to the tube. And then I'll be able to extract, I'll be able to clean up the beads and wash them off and clean them up. And then finally I'll take the tubes off the magnetic rack and put some water in there to resuspend the DNA that I want from off of the beads. We'll let that go for 5 minutes. Can you see it? The solution is clear now. There's little pellets right there. All right, so now that it's pelleted, I'm going to pull off the liquid and leave the brown magnetic beads in the tube where my DNA is bound to. And now I'm going to wash it with ethanol, 200 microliters. Let it sit for 30 seconds in ethanol. I'm going to leave it on the stand so the beads stay bound in the tube with the DNA. And then I'm going to take the ethanol out. And then I'm going to do an extra wash of ethanol because I'm trying to target something specific in my change to the protocol. Wonder what that could be. And now I'm going to take out all the ethanol. And I want to make sure I get as much as possible so that I don't carry over any ethanol to the next step when I resuspend my DNA and get it off the magnetic beads. Because ethanol is just going to mess everything up later if I leave it on there. Get all the ethanol out, and I'm going to leave it on this, and I'm just going to let it dry. Let all the ethanol evaporate for 5 minutes. I don't want to let it dry too much or else the DNA will really get stuck to the beads and I'll never get it off. But I'll let it dry for 5 minutes so that all the ethanol disappears. So now you can see that there's a little pellet. It's all nice and dry. I'm going to add 50 microliters of TE. I'm going to take it off the magnet so that I can resuspend the beads in the TE. And you see that it kind of sticks to the side. So what I'm going to do is I'm going to go up and down a couple times to try to wash it off.
For 2 minutes outside of the stand.
Yep, and then I'm going to let it sit outside the stand for 2 minutes to allow all the DNA to elute off of the beads. There are the beads again. Resuspend it so it's nice and even, and let it sit for 2 minutes to get the beads, the DNA off the beads. All right, so after 2 minutes, I'm gonna put it back onto the magnetic stand, and I'm gonna pellet the beads. And the DNA is now no longer on the beads, bound to the beads, it's now in the solution. So now I'm gonna pellet the beads, and I'm gonna suck up the solution, and I'm gonna keep the solution, because the DNA is inside that, and throw away the beads. You can see it's already pelleting. Take about 2 minutes. I still got like 20 seconds. Make sure you don't carry over any beads when you pull out your liquid. Sometimes to avoid that, I'll take less than, I put in 50 microliters, and I might try to take out less, like 48, 47, 46, so that I don't pull over any beads. All right, so now I'll try to take it out without disturbing the pellet. So just like before, I try to take the liquid out on the side of the tube away from where the pellet is, so I don't take any of it with me. There we go. Isolated DNA, ready to be assessed with nanodrop, qubit, and run on a gel. I think this is these are the winning tubes right here. So we're going to be using this qubit machine over here, it's a fluorometer, to get a really precise measure of the amount of DNA that we have. The nanodrop gives you a rough estimate, but unfortunately the spectrophotometry method is really susceptible to being incorrect if there are contaminants in your sample. Things like ethanol or salts or proteins. This fluorometer works a lot better. And so what it does is that we've created a master mix solution here that's got buffer and a dye in it. And the dye will bind to the DNA. And it's a fluorescent dye that only binds to DNA. There's a different one where the fluorescent dye only binds to RNA. But because the fluorescent dye only binds to DNA, and then when we put it in this fluorometer, it will measure just the amount of DNA that this fluorescent dye is bound to. So it's a very precise measure for us figuring out how much DNA is in it. So we're going to run a couple standards, and then we'll run each of these samples through and see what the actual yields were of our DNA isolations. Tubes are always clean and positioned in the same way each time we use them. We're actually using special tubes that are designed just to use in this machine. We're going to load the tube in. We're going to hit double-stranded DNA. We are using DNA broad range, so we're going to hit that. First thing I'm going to do is read the standards in. So I've inserted standard 1. I'm going to read standard 1. So that's my initial point. I'm going to put it back in. I always have the hinge pointed to the back. And I'm going to read my second standard. And as you can see, I've now made my standard curve.
So it goes right here. And then I put down the arm and I click measure. And it'll give you a curve like this. So we can see right here is the amount of nucleic acid, and I've selected DNA, so 75 is not bad. And then we can see our ratios here. 260, 280 is at 1.93, which is pretty good. We want something close to like 1.8 or 2. And then our 260, 230 here is 2.32, which isn't bad. We want something around 2 or 2.2. So overall, this one's all right.
So we've made our standard curve. Now we're going to add in our sample to each of these tubes. We'll then mix it up, clean it off, put it in with the hinge towards back, and read the tube. So here it tells us that our sample was actually 73.4 nanograms per microliter. So we'll write that in the notebook. Qubit 1, 2, 3, 4, 5, 6. 73.4 nanograms per microliter. Notice that that's a lot less than what we had thought was in there based upon the nanodrop.
Okay, hi everyone. Now that we've extracted our DNA, we need to quantify it and check the quality of the DNA. So to do that, we're going to use this machine here called a nanodrop. It gives us sort of a rough estimation of how much DNA we have, but more importantly, we can check the quality. So we check two ratios. The first one is called 260-80 ratio, which gives us a sort of idea about how much protein contamination we might have in relation to how much DNA we have. And then we also have a 260-30 ratio, which tells us if we have any ethanol or salt contamination. So I'll just load our sample here. I'll put 2 microliters onto this core on the machine.
So we are going to load the gel now to check the weight of our samples to see which sample has the greatest molecular weight and therefore has the longest chain. I'm going to load a ladder in the first well so that we can compare the standard, and then the next 6 samples will load 20 microliters of the sample.
So now we're going to run this gel at about 110, 115 volts for about 30 minutes or so. So we can get a nice spread out on the gel, and we'll use the dye to track to make sure that it doesn't run too far. So gel finished. Looks pretty good. This is a marker of how far we think it's run, or at least part of its run. Usually we might want to shoot for something maybe a little bit farther down, but let's see what it looks like. So we're going to put this under a transilluminator. This is a gel dog. You guys we used a different version when we did the labs before. This is a fancier version, but does the same sort of thing. We'll pick the right tray right here for the stain that we're using. This is the one that you use for cyber stains, and that's what we use for this gel. You can also use this for gel red, which is what we've used in class before. And all we do here is we just take the gel, we slide it onto the drawer. We then close the drawer, close the gel. Make sure the image is centered. And then we're going to select which application we're going to do. We're doing nucleic acids. We're not doing gel red. We're doing cyber safes. So make sure I hit that button. And then after that's done, I just hit the camera button. It's adjusting the exposure or the brightness of the gel. And then this is our final gel picture. What we can see here is there are smears across these lines. What we're targeting, right, is high molecular weight DNA. So we want to figure out which one of these we think has the most and darkest bands up here and the lightest, faintest bands down on this end. Some of these large smears that we see here that are very short could very well be RNA carryover. Especially if, say, we look at the example here for sample 1, which is, I'm assuming this is sample 1 right here. This is something that Nanodrop said we had a lot of DNA. Then when we did Qubit, it said we didn't have anywhere near as much of that DNA. That's probably because it picked up a lot of RNA. And that's what it's observing right there. So overall, it looks pretty good. I'm going to take the image. I'm actually going to transform it a little bit. I'm going to invert it, because inverted gels just look easier to read and they print better. You can also, I might try to lighten it a little bit. And now I'm going to save it as a JPEG. All right, I'll put it up on the island side and you guys can examine it yourselves. Go through and rank which samples you think are the best, 1 through 6. All right, so I brought all the sequencing stuff to my house. So now I've made a little molecular lab at my in one of my rooms, and I'm going to do the sequencing here. So first thing I have to do is I have to do a library prep, and it's a really simple one here. As we talked about in the intro lecture materials, basically we use a really simple one-step library construction where we use a transposome to take a complex. It's a protein complex with some primers attached to it. And every time that protein complex runs into a piece of DNA, it's going to ligate two primers, like right on the end of that piece of DNA. And then we're going to add in a little sequencing tether to the end of that. And that tether is going to be the thing which helps the piece of DNA migrate to the nanopore and then go through the nanodrive and pull the DNA sequence right through that nanopore so it can get sequenced. So, real quick right now, I'm going to build these libraries. We've got our 2 little samples. Give it a little flick to mix it. Get all the stuff down at the bottom. Good to go. We're going to do a quick incubation using something called a mini PCR, which is just a tiny little thermocycler, which I brought home. And if you look inside of it, you can see our 2 little samples in there. So now, it's a very quick little protocol. All right, now I'm going to add some magnetic beads to bind up my library and to clean it up. An equal amount to each of these tubes. So 10 microliters and 5 microliters. And then I'm going to let it mix for 5 minutes on the hula, or the nutator. All right, let's see how it goes. Got the little PCR machine, the little thermocycler running. It's doing a 1 minute incubation at 30 degrees C, and then it'll do 1 minute at 80 degrees C. All right, well, we've got both of our MinION sequencers going right now. Here, this one is running a regular MinION sequence, and this one's running a Flongle, which is going to make less data, but is a lot cheaper. These are 2 little minutes. They are graphic processing units, like you use to play your video games, that are actually doing the base calling. This is one of the computers, which is attached to 1 minute, showing the sequencing that's going on. And you can see here how many sequences have been put out and what their size distribution looks like. So right now, it's just been running for a little bit, and we've already made 23 megabases of data, about almost 30,000 reads. This is the Flongle. It's got a lot less flow cells. You can see it's making less sequence, so it doesn't make as much sequence as the other one because it's a smaller flow cell. So we got our sequencing going, and hopefully everything will look good tomorrow. Got the little PCR machine, the little thermocycler running. It's doing a 1 minute incubation at 30 degrees C, and then it'll do 1 minute at 80 degrees C.